Okay, I think we can start. So it's a pleasure for me today to introduce Dario Greco. So I don't know for the people who don't know him, Dario is a professor of bioinformatics at the Faculty of Medicine and Health Technology at Tampere University, and also the director of FAVE, which is the Finnish hub for development and validation of integrated approaches. As well as professor of pharmaceutical bioinformatics at the University of Helsinki, a principal investigator at the Institute of Biotechnology in Finland, the University of Helsinki, as well as coordinator of the Finnish 3R Center. Dario published more than 200 peer-reviewed articles, book, chapters, reviews in the areas of nanotoxicology, toxicogenomics and drug discovery, network biology and data modeling, and bioinformatics. And as a principal investigator, he received funding from the Academy of Finland, the European Commission, the Novo Nordisk Foundation, and the Finnish Red Cross and Finnish government. More important, he is part of many ongoing projects. He leads, for example, the European project Archimedes, which is part of the ERC Consolidator Grant, which focus on developing in vitro and computational NAMs and IATAs for pulmonary fibrosis, as well is leading the OECD project Omics to AOP. In addition, Dario is also coordinator of the European project Insight and of the TULI project funded by EFSA, as well as the deputy coordinator of the Chiasma project, which tries to focus on development on the development of integrated, safe, and sustainable biodesign framework and the development of NAMs to replace animal experiment in biomedicine, toxicology, and pharmacology. Today, I will be speaking about multi-scale modeling. So Dario, the floor is yours. And just for all the participants, this meeting is going to be recorded and will be available on the EUROTOX YouTube channel. Thank you.
Great. Yeah, thank you very much for having me here today, and thanks so much for the kind words and introduction. So yeah, indeed. So today I will focus on multi-scale modeling, and I will describe a little bit of the work we are doing, especially in trying to bridge toxicology with other biomedical research fields, especially with the epidemiology. So let me start with something that is probably obvious to many of us in this audience, that one of the greatest facts that we have witnessed in the last decade or so is an explosion of the chemical universe known to us. So we went from having a chemical universe of a little bit over 700,000 chemicals in the early 2000s, and nowadays the number of chemicals that we know or can be synthesized well exceeds the 40 billion. This, as we probably know, brings two paradoxes. So the first one is that obviously we have the ability to test thoroughly only a limited number of chemicals that are put on the market. What is more striking to me personally is that the estimation is that we have a new chemical emerging from R&D every 4 hours on a daily basis. On the other hand, we have the paradox of the drugs. So we know that drug development is much slower than we wish for. It's still extremely expensive, and we know that most of the drugs actually fail to reach the market for safety reasons. Now, toxicology in its most traditional form focuses on defining safe doses, the reference doses from animal experiments, or also more recently with using in vitro models or even computational models. But traditionally, toxicology focuses on the analytical endpoints of the exposure. And in this paradigm, essentially, because we don't resolve any of the mechanisms, we learn very little of what really is happening behind in the biology and behind the final phenotypic endpoint. And so toxicology is facing a massive evolution at this stage. So we are shifting from focusing only or mainly on phenomenal facts to actually describe more and more mechanisms. So we really want to define the whole cascade of events that occur when a chemical gets in contact with the biological system. And so in order to do this, there's an emerging complementary approaches in toxicology that I like to put under the umbrella of systems toxicology, where we all focus on gaining basic information, for instance, by the use of omics technologies and others, that can be used not only to define the safe conditions in which a chemical or a drug can be used, but also importantly to gain knowledge that can be used to design next generation chemicals and drugs. But we have to acknowledge, and this will be a main theme in today's talk, is the fact that whenever we study the interaction between chemicals and biological systems, we need to do this in the context of a multi-scale interaction. So obviously the chemicals interact with biomolecules at the molecular scale, but these interactions eventually have trigger a cascade of events that have obviously effects at the level of the cells, which in turn trigger a cascade that has an effect on the level of the tissues and organs, which eventually then become visible at the level of the individual, so at the clinical level. Interestingly, in toxicology, we have seen the emergence of a number of models in vitro and computational that address specific questions regarding specific scales of this multi-scale interaction. And so what happens is that the majority of the models we have developed, including the vast majority of the NAMs, actually are set to look for associations between either characteristics of the chemicals or characteristics of chemicals and characteristics of the biological system in order to predict certain toxicologically relevant endpoints. They work as black box. And we definitely need to gain mechanistic information, which eventually means that we have to go deeper in the understanding of how chemicals interact with the multi-scale of our biological systems. This is also that even toxicogenomics approaches as used in the vast majority of the studies in the last decade are not enough, because we describe the interactions of the chemicals with biomolecules, but we don't necessarily describe the interactions of the biomolecules with each other. And we know that this is fundamental because all our cells are virtually equipped with the same genes, and different ways of interacting of the genes and proteins and the different molecules that compose our cells actually define functions, emerging functions that we call tissues or organs. We know that these higher structures also interact with each other, defining specific individuals. But interestingly, this game can be done even beyond the individuals, because we know that humans don't live in isolation. We live in a rich environment. We entertain a number of functional, for instance, alimentary interactions with other species. So why not to look at the patterns of interactions at different scales, so that we can unify our modeling approach in our investigation to focus on what I like to call the fabric of life. So how do we do this? Well, essentially we have three main approaches that are emerging in the field. So the first one is by integrating data that address different characteristics of this chemical biological interaction at multiple scales, by integrating models that are by themselves not mechanistic nature, that can be combined to answer specific questions in a multi-scale, so become mechanistic in this way, or then anchor our investigation directly on a mechanistic framework, like for instance the adverse outcome pathways. So the first step is integrating data. And this is emerging in the field in the form of knowledge graphs. These are data structures that they can be very large, where essentially the nodes, the networks, the nodes are data points or concepts, and the edges that connect the nodes are somewhat a level of relatedness or causal relatedness between the data. For instance, this is the simplified structure of the knowledge graph that we have developed. We are still developing in my group, which at the moment is comprising over 7 million data points from multiple data sets and databases. So, for instance, we can use now knowledge graphs in combination with machine learning algorithms to answer specific questions. This is an example of an ongoing project in the group where we built a knowledge graph of a little bit over 45,000 nodes and a bit over 1 million edges, where we ask specific questions on what kind of human diseases the phthalates can cause. Now, when we look at the black box, so to say, association of phthalates to human diseases, we find no surprises here. So the diseases that we predict being associated to phthalates are obviously related to endocrine disruption, to developmental abnormalities, and so on and so forth. But because we include in our knowledge graph also mechanistic information in the form of key events and their molecular anchoring, then we are able also to retrieve from our predictions potential cascades of events that from the molecular through cellular systemic scales then might link specific phthalates to specific diseases. Here you see the example of the connecting points in the knowledge graph we built that connect specific type of phthalates to cryptorchidism. Now the second stop is integrating models, as I said. And here the beauty is the fact that we can reuse models in vitro, computational, that are not mechanistic in nature, but that can be combined to towards a mechanistic understanding. And this is, for instance, the work that the whole European Consortium Insights that I have the privilege to coordinate is doing. So basically here we identified a large collection of especially computational models that's answer to specific questions related to chemicals and their interaction with biological systems at different scales. So you see, for instance, the token, the QSER mainly answer to questions related to molecular, maybe cellular. We have, of course, the high-throughput screening, we have PBK, which is more on a tissue and systemic level, and so on and so forth. And so this is, for instance, an example on how we have integrated PBK with toxicogenomics modeling coming from in vitro models of in vitro liver, and how this system reacts to different types of PFAS species. So you can see here, these are specific triggering doses that have been predicted by PBK modeling, and then we can use now this information to adjust the concentrations that we screen in vitro towards finding specific cascades of key events with their own specific safe dose or kind of reference dose, which eventually form the mechanism of action of PFAS on the lung, and here, not surprisingly, pointing to liver fibrosis. I keep telling lung, but it's liver so far. So stay with me. Now, the third approach is mechanistic modeling that is native in mechanistic nature. And so here we specifically focus on the use of AOPs, adverse outcome pathways. I'm sure these are quite familiar to many people in the audience. So these are multi-scale cause-effect models that explain or kind of link exposure to specific stressors to certain adverse outcomes. Interestingly, also the adverse outcome pathways form a network because a number of key events are actually shared between different AOPs. The nice thing about the AOPs is that they are also regulatory friendly in a way, strongly endorsed by the OECD. So that means that putting our analysis in this framework helps us also to be more regularly relevant in a native manner. Now, the problem with the adverse outcome pathways is that they are defined as theoretical semantics. So they are concepts, essentially. And so the problem is that we need to anchor these conceptual key events in the AOPs to something that we can measure. And so towards this end, we recently made quite huge effort in terms of annotation of sets of molecules that can be associated specifically to key events in the AOP framework. We did this by using language model, computational language models, followed by a massive amount of manual curation. And so now we can start anchoring these concepts to something that we can measure. And we can do also a number of interesting things. For instance, in the beginning of the pandemic, by using our molecular annotations of VOPs, we could predict that pulmonary fibrosis could be a long-term effect of the acute COVID-19 disease, because we found a number of interesting commonalities and similarities between what was known at that time as the COVID-19 AOP and an AOP that is kind of quite close to the heart of the particulate matter toxicologists, which leads to also pulmonary fibrosis. Another way we can exploit now this mechanism multi-scale system is, for instance, by adding information on what is the specific applicability domain of NAMs that we may use. This is important because NAMs traditionally have been developed just with the aim of mimicking specific pathophysiological characteristics of certain organs or tissues, without let us understanding exactly which mechanisms could be essentially observed by in vitro models. So when we put these now in the context of this multi-scale mechanistic modeling framework, then we find this is an example we published a few years back where we had a number of nanomaterials that were investigated by transcriptomics in vivo in the lung of mice exposed to these materials, as well as in two in vitro models, human in vitro models. And here you see this is the altogether the number of AOPs and key events that can be observed in one or multiple of these. And you can see that there is a lot of the mechanism that indeed is overlapping between the in vivo here in orange and at least one of the two in vitro models. But there are also certain mechanisms, like for instance, this down here, this here, that we cannot observe in vitro. And this is something that we expect. So in vitro obviously does not capture all the complexity of the whole organism. And again, another way how we can use this approach is to define an alternative way for grouping chemicals. We know that chemical grouping is one of the big themes in nowadays toxicology, and traditionally this is done by looking at either at toxicological endpoints or at structural or chemical characteristics of the compounds we're studying. He proposed a new way of doing this by looking at the mechanism of action that would reconstruct for each of the chemicals we included in our study, as in terms of which cascade of key across the scales we could reconstruct from each of these. Ultimately, we can also go beyond human health towards One Health approaches. So by using the same mechanistic multi-scale modeling, we could reconstruct a common mechanism of action, regulatory mechanism of action, which in this case goes into the epigenetic response of different organisms across the tree of life to nanoparticulate exposure, is indeed common to multiple species, and it's also explaining a relatively large amount of the mechanism of action that we observe in each of these species individually, although when we look at the level of molecules that are directly affected in each of these across nanomaterial exposures, then they are extremely different from each other. Now, as I said, so toxicology is obviously very into the future and predictive in nature. And kind of in a way, I'll put it in as a complementary approach to epidemiology, where we look in the past and we check when maybe a damage by an exposure has already occurred in the wildlife. And I like to think of systems toxicology approach as kind of a nice bridge between traditional toxicology and traditional epidemiology. So again, here, where is the idea is that toxicology is looking, obviously starting from the chemical as a focus point, so starts from the molecular scale and then trying to predict the effect across the scales, while epidemiology obviously looks at the things that have already happened. So in this case, then we start from the individuals and their clinical characteristics, and we go down in the prediction of the scales. And this is exactly the exercise we did when we compared, so we integrated together omics-derived signatures for over 1,600 chemicals in 6 different species, including human. And then here we focused on a large collection of EWAS studies. So basically DNA methylation, genome-wide DNA methylation data where we have association between CpG methylation levels and over 650 different traits, human traits across multiple tissues. So here the idea is to map now the epidemiological data from the clinical side, from the clinical scale, and predict down, and instead start from the molecular omics derived signature of the chemicals and kind of enter the multi-scale from the molecular side and then predict up. So we did a little bit of algorithmic development to allow these, and this is, for instance, one of the results we retrieved. So we started to look at signals coming from EWAS or from chemical data would join across the same AOP pathways. And for instance, this is an example linking titanium dioxide exposure with an epigenetic signature that we retrieved from air pollution exposure to the blood. And here you see a number of interesting themes that we kind of can expect. So ROS production, DNA damage, airway activation, and so on and so forth. More specifically, what we are able to do is to focus on specific patterns of molecular and epigenetic regulation that underlies this mechanism. And so here we identify also a number of DNA domains and their putative binding site ability for specific transcription factors that might orchestrate this connection. The last point I wanted to address today is how do we go beyond traditional toxicology, in the sense that toxicology is funding as a field. It's clear that nowadays it's not anymore enough to look at human health or environmental health. We really have to consider a number of multiple impact dimensions beyond and above traditional toxicology. So for instance, looking at social and economic impact dimensions towards safe and sustainable by design paradigm. And so in this sense, we started to look at the possibility of building chains of mechanistic events that could explain in a mechanistic and multi-scale fashion also social and economic impact of chemical exposure. So we started from specific sets of scientific literature in the LCA, specifically in the life assessment field, and we extracted with the use of language models computationally. We extracted a number of putative social and economic key events, and then through again a quite thick amount of curation, then we distilled a number of what we call impact outcome pathways, so chains of events that don't lead to human health endpoints or ecotox endpoints, but to social and economic endpoints. And then we mapped these new chains of social and economic pathways to the AOP framework. And so we started to observe, for instance, something interesting. Obviously, many things are also quite well imaginable or known. For instance, chains of key events that lead to decreased biodiversity have a strong link to sensitization in humans. We know that, for instance, our kids who live in a less rich biodiverse environment, then they develop asthma more often than kids in the countryside. We know also, and something else that we found is that mechanistic chains of events that leads to lower socioeconomic status then works extremely strongly as a modifying factor towards an increased level of basal inflammation, which we know is linked to a number of diseases, and we have learned that this was also a major factor in during the COVID pandemic. So let's conclude here this brief kind of walk through some of the things we're doing. I want to leave with this home message. So traditionally, we looked at phenotypic apical endpoints. And at this level, whether we observe this by the use of animal models or in vitro models or computational models, the chemicals look pretty different from each other. So we learn little about kind of One Health or SSPD. The more we add mechanistic information to our investigation by the use of more advanced alternative methods, so the NAMs, and integrating multi-scale data and computational models, then we start observing interesting commonalities between modalities or mode of actions of different chemicals with each other, which eventually leads to what we hope for the next future, to be able to gain enough information that we can really use for SSPT development. So we'll close here today, and I would like to thank, of course, the amazing group of people working with me here up in Finland, in Tampere, and Helsinki, of course, our funding agencies and networks we are involved with. And I will leave with the notion that we are currently hiring. So if any of these I spoke today resonates with you, or if you know someone who might resonate with this stuff, then please don't hesitate to contact me later. And of course, if there is room for questions, I'll be happy to take those. So thank you much.
Thank you very much, Dario. Very interesting talk, and I think it generates a lot of forward thinking about where we are heading with all the amount of data and the advancement that we have in the area of generative models and AI. So I was checking in the box if there are questions. It looks like I don't see any, and maybe we can start. I have one for you. So the first one I have for your talk is I'm wondering because we have a lot, very massive amounts of data. So I'm thinking, what are the bottlenecks in terms of either on one side, I would say maybe computational bottlenecks when managing and handling all those data, as well as, for example, you mentioned before about using AOPs to do AOP-based grouping. We know that there is a lot of currently. I mean, if we look at the AOP, it's quite limited. There is a little bit of gaps for some of endpoints. How do you see? I know that there is a lot of, active, a lot of activity in developing new IOPs. So probably this is something it will improve in the future. But what do you think in terms of bottlenecks when you look at those kind of
So
models?
yeah, I mean, interesting points. Thanks a lot. So the first one is computational bottlenecks or data bottlenecks. So the biggest bottleneck we have currently in data is the quality of data. So while, for instance, curating large amount of toxicogenomics data in my group, we realized that in average, 1 in 3 data sets, although perfectly fair, should have never been used. It could not be used because of massive flaws in the experimental design. So, and obviously these are very sensitive systems too, especially when we speak about more advanced machine learning or AI models. They are very sensitive to data quality. So if you put garbage in, you get garbage predictions out. That's the big problem. So bottleneck at the moment is we have to look as a community beyond concept of fair principles and understand that there is a specific type of evaluation that we have to do on the quality of data, which of course is intrinsically helped by the fair principles. Now, to the second of your point, AOPs. Yes, indeed, there is a lot of work to be done. And there are multiple massive efforts for defining new AOPs, especially in areas where we don't have many AOPs or we have none. So a lot of work needs to be done. And what I have shown today must be taken as a point of proof of concept. Yeah. So still a lot of work to be done in AOP development there.
Okay, thank you very much. And I see there is a question from Jacob. Jacob, can you post it in the chat? Because I cannot see it. I don't know whether you post it. Otherwise, please, so I mean, I cannot see it, but please unmute yourself and ask the question if you want.
Thank you for the privilege. My name is Jacob from Nigeria. Can you hear me?
Yes.
Okay. In your adverse outcome pathways analysis, it is quite an interesting topic for me, and I've been trying to see how I can get myself and understand this topic very, very much. So I want to ask, how do you get your, I think, what are the databases you use in getting your data? Because most of the published database, so most of the published data are not reliable to some extent. And what are the database, and as in, do you have any reliable databases that you get your data? And now when you get your data, how do you validate it so that it will not tilt your prediction, or it will not give a wrong prediction? So that is, and then what is a scaling approach? What is your scaling approach to your data? Thank you. That's my question.
Thank you very much, Jacob. Yeah, really interesting question. So let's start from the beginning. So databases. So when it comes to AOPs, so the reference database for AOPs is the AOP Wiki. So that's the place to look for AOPs in their most trustworthy way. Now, those are concepts. So we had, and there is no molecular annotation to my understanding except ours at the moment published. So we had to do it ourselves. When it comes to quality of other types of data, for instance, omics type of data or other types of data, then there is not at the moment, unfortunately, there is not a database which is more trustworthy than another. So that means that whenever working with publicly available data, you need to add a very careful layer of data quality, data check, and data curation. That's what we do in our routine, and that's what I advocate always. To the third point, the validation. Yes, indeed, you're right. So when it comes to the molecular observations that or predictions that we make, we do validate our models in vitro in the lab. So we do that by generating new data. When it comes to other types of data, it's a little bit more complicated. But nowadays there is enough of public data out there that you can use also to validate your predictions. So that's my recipe for the time being.
Thank you, Dario, for this answer. So I don't see additional question, but since we have more time, if you don't mind, I will have a couple more for you. So the first one is really about the fact that we have a lot of novel chemicals. So I'm wondering whether multiscale could generalize for novel chemicals which are, let's say, outside of the training or the experimental space. And linked to this, whether you see multiscale modeling like becoming more than predictive first rather than mechanistic first because of that.
That's a good point. So I like mechanistic first more than predictive first, simply because I mean mechanistic first allows you to explain what you're predicting, right? Now, there is no doubt that the applicability domain in whatever we are doing, even in the mechanistic predictive modeling, is fundamental. What is beautiful of the multi-scale approach is that if you deal with a new chemical that is absolutely outside any uh, applicability domain, because you are having a mechanistic multi-scale modeling approach, you can start measuring something in one of the scales. And for instance, nowadays generating omics data in vitro is fairly simple, quite cheap, right, for most of our labs. And then starting from there, then you can start predicting what would happen to different scales. This is also my specific recipe to decrease the number of animal experiments, right? Because if you look at in vitro data just in the context of in vitro data, you will still need animal models to capture systemic effects. If you put in vitro generated data, not only omics, but also any other type of data in a multi-scale modeling approach, then you can eventually make more better decisions and refine, if not replace, but refine at least your animal tests, right?
Okay, thank you. And then, I mean, I have a last one, which is probably you, you received this question many times, and I wouldn't even call it like regulatory acceptance, but more regulatory recognition. So it's just more a thinking because with multi-scale modeling, basically what you're doing, you are merging a lot of data that are coming from many methods, which themselves, they are not validated or qualified. So now you are kind of using all of them, and how do you see this going forward for regulatory recognition, approval, or uptake? Or do you think that maybe the benefit is truly that the fact that you have many findings, and so in terms of weight of evidence, it will give you a little bit more, so it will reduce the uncertainty on the findings, you know, so what I mean?
That's a really good point. So first of all, we are not yet there, but we are closer than we were a few years back. So that's the good news. Now, yes, you're right. The more stuff you bring together, the bigger weight of evidence you have. And that's a good starting point. The other point is also to bring the discussion among the regulators and industrial parties as well, to really embed the multi-scale mechanistic modeling as the way to go. And this is, for instance, something that we are actively doing in the context of this omics to AOP project, which is OECD, an OECD project. I have the privilege to coordinate it together with the ECFAM, with the JRC. So in there, that's exactly what we are doing, trying to standardize this multi-scale mechanistic modeling as much as possible so that we can go in that direction. But this is not a solution that we have for next week. So there is still work to be done, obviously.
Okay. Thank you very much, Dario. And I have a question from Aditya Singh in the chat. Is asking basically, given that FAVE focusing on developing YATAs, how can we formally quantify the uncertainty of AI predicted relationship in a knowledge graph to a level that satisfies the rigorous validation criteria required by regulator agencies for replacing traditional in vivo studies?
This is an excellent question, because knowledge graphs have a fundamental bias that obviously, because you include the data that supports certain connections between your nodes in the graph, the false positives are easy to estimate. The problem is that you don't know whether a missing edge from the original data is really an edge that shouldn't be there, so the A and B are not connected, or it's simply missing observation. And this is a fundamental problem in those who work with predictive models based on knowledge graphs. Now, We have some tools, specific tools in the field of toxicology. For instance, when we speak about drug safety, we use a lot models that rely on the absence of connections between drugs based on drug interactions, for instance. But we still need a lot of data to be generated in order to resolve this problem at a large scale. So a lot of work needs to be done.
Okay. I have another one still from Jacob, so if you want to answer, since we have time. So he's asking which large language model do you use or will you recommend in predictive toxicology, whether there is a point where you would use more than two to predict. So in one prediction, let's say more than models, so more than two models.
Yeah, that's an excellent question again. So my blunt answer to which LLM model is better for predictive toxicology, the answer is none. LLMs cannot be trusted by definition. Their reproducibility is basically non-existent, and even like an ensemble approach where we try multiple of them is badly failing at the moment. This is not a bias or a fault in the development of LLMs. It's actually how LLMs are built. This said, LLMs can still be useful when, for instance, we don't ask specific or direct questions towards predictive toxicology, but for instance, we embed them into pipelines like those that I showed today, where we use language models to extract concepts from the literature, and then maybe we do manual curation to confirm them. So in this sense, LLMs in toxicology are becoming very useful. There is an enormous amount of not only data, but really knowledge to be mined from not only from the scientific articles, but also let's think about everything we know from the regulatory agencies and all the reports from regulatory agencies. So in that terms, that's fine. My specific policy in my group, and I know that my students are suffering a lot because of that, is whenever you put an LLM for whatever you're doing, then you need to do a manual curation afterwards. So don't trust LLMs alone. They are not tools for doing predictions, really. Yeah.
Thank you very much, Dario. I don't see any other question. So I think we can close it here. Thank you again for your talk. And as I mentioned at the beginning to all the participants, so the recording will be available on the EUROTOX YouTube channel for you to look. And thank you very much for being here.
Thanks so much for having me. Have a good day. Take
Thank
care.
you. Bye.
Bye.